Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. We are from Group 5. We will explain about plant defense. Next. There is our member. First is Syahdani Izzudin. Second is Siti Ihwani. And then Sesilya Nur Oviarsya, Sely Kikiba Putra, Bintang Prakosa Jati, Rizky Maulana Putra. And last is Awal Rahmat Marwangsa. Next. So each plant species is affected by approximately 100 different kinds of fungi, bacteria, molecules, virus, and nematodes. Frequently, a single plant is attacked by 100,000 and in leaf spot disease of large trees, probably hundreds of thousands of individuals of a single kind of pathogen. Next. In general, plants defend themselves against pathogen by a combination of weapons from two arsenal. First is structural defense, is defense mechanism that act as physical barrier and inhibit the pathogen from gaining entrance and spreading through the plant. And second is biochemical defense, is defense mechanism that take place in the cell and tissues of the plant and produce substances that are either toxic to pathogen or create conditions that inhibit growth of the pathogen in the plant. Next. Both structural defense or biochemical defense can be characterized as first is pre-exciting, is defense mechanism in plant to prevent an infection. And second is post-infection, defense mechanism after the plant has been infected.
Structural defense. Point first, pre-exciting structural defense mechanism. Cuticular wax. Waxes on leaf and fruit surfaces from a water repellent surface, thereby preventing the formation of a film of water on which pathogen might be deposited and germinate or fungi or multiple bacteria. Thickness of cuticle. A thick cuticle may increase resistance to infection in disease in which the pathogen enter its host only to direct penetration. Next. Structure of epidermal cell wall. The thickness and tautness of outer wall of epidermal cell are apparently important factor in the resistance of some plant to certain pathogen. Thick, tough wall of epidermal cell make direct penetration by fungal pathogen difficult or impossible. Structure of natural openings. Some pathogen enter plant through opened stomata, like the stem rust of wheat. Thus, some wheat varieties in which the stomata open late in the day are resistant because the germ tubes of spores germinating in the night dew desiccate due evaporation of the dew before the stomata begin to open. Next.
Structural defense. B. Post-infection structural defense mechanism. Cytoplasmic defense reaction. The cytoplasm of the invaded cell surround the hyphae of the pathogen and the nucleus of the host cell gets fractured to break into two. In some host cells, the cytoplasm and the nucleus of the infected cell enlarge. The cytoplasm become granular and dense and develop granular particles. This results in the disintegration of the pathogen mycelium and the infection stops. Next. Cell wall defense structure. Three main types of cell wall defense structure have been observed when disease. One, first, the outer layer of cell wall of the parenchyma cell in contact with invading bacterial cell produce an amorphous fibrillar material that trap the bacteria, thus preventing them to multiply. Second, cell wall thicket in response to the pathogen by producing a cellulose material, thus preventing the entry of the pathogens. Third, cellulose papillae are deposited on the inner side of cell wall in response to infection by fungal pathogens. If papillae are present before inoculation, they also seem to prevent the pathogen from subsequently penetrating the cell. Next.
Histological defense structures. The following four developments take place in the tissues after penetration. First, formation of cork layers. Infection pathogens frequently induce plants to form several layers of cork cells beyond the point of infection, apparently as a result of stimulation of the host cells by substances secreted by the pathogen. Second, formation of abscission layers. Abscission layers formation protect the healthy leaf tissues from the attack of the pathogen by discarding the infected area along with a few yet uninfected cells. Protect the rest of the leaf tissue from being invaded by the pathogen and from becoming affected by the toxic secretion of the pathogen. Third, formation of teloses. Teloses from in xylem vessels of most plants under various conditions of stress and during invasion by most the xylem invading pathogens. Teloses have cellulosic walls and may, by their size and numbers, clog the vessel completely. Fourth, dissipation of gums. Plants produce a variety of gummy substances around lesion or spot as a result of the infection. These gummy substances inhibit the progress of the pathogen. The gummy substances are commonly produced in stone fruit.
Next, necrotic structural defense reactions. Defense through the hypersensitive response. In many host pathogens combination, as soon as the pathogen establishes contact with the cells, the nucleus moves toward the invading pathogen and soon disintegrates. At the same time, brown resin-like granules form in the cytoplasm, first around the point of the penetration of the pathogen and then throughout the cytoplasm. As the browning discoloration of the plant cell cytoplasm continues and death set in, the invading hypa begin to degenerate. In most cases, the hypa does not grow out such cell, and further invasion is stopped. Next is biochemical defense. This part consists of pre-existing biochemical defense mechanism and post-infection biochemical defense mechanism. Next. Pre-existing biochemical defense mechanism inhibitor release. Inhibitor released by the plant in its environment. Plant extract a variety of substances throughout the surface of their above ground part as well as through the surface of their roots. Some of the compounds released by a certain kind of plants seem to have inhibitory action against certain pathogens. Inhibitors present in the plant cell before infection. Some plants are resistant to disease caused by certain pathogens because of one or more inhibitor antimicrobial compounds, known as phytoanticipins, which are present in the cell before the infection.
A lack of essential factors. First, lack of recognition between host and pathogen. If the pathogen does not recognize the plant as one of its hosts, plant it may not become attached to the plant or may not produce infection substance. 2. Lack of host receptor and sensitive site for toxin. Only plants that have such sensitive receptor or site become diseased. Plants of other varieties or species that lack such receptor or site remain resistant to the toxin and develop no symptoms. 3. Lack of essential substance from the pathogen. Species and species or varieties of plant that for some reason do not produce of one of the substance essential for the survival of an obligate parasite or for development of infection by any parasite would be resistant to the pathogen that requires it. Post-infection biochemical defense mechanism. Post-infection biochemical defense mechanism. Phenolic compounds that are toxin to pathogen are produced and accumulate at a faster rate after infection, especially in resistant variety of plant relative to susceptible variety. Chronogenic acid, caffeic acid, and ferulic acid are examples of such phenolic compounds. Phytoalexins. Phytoalexins are toxic antimicrobial substances produced in appreciable amount in plant only after stimulation by various types of phytopathogenic microorganisms or by chemical and mechanical injury. Phytoalexins are produced by healthy cells adjacent to localized damage and necrotic cells in response to material diffusing from the damaged cell.
Number 3 is substance produced in the host to resist enzyme produced by pathogen. Some hosts produce semi-chemical which neutralize the enzymes produced by pathogen. In bean plants, infection with Rhizoctonia solani causes necrosis. In resistant bean varieties, the entry of pathogen causes the separation of methyl group from methylated lactic substance from polyvalent cation of lactic salt which contain calcium. Number 4 is the detoxification of pathogen toxin and enzymes. In some cases, the plants produce a chemical which deactivate the toxin produced by the pathogens. For example, Piracularia oryzae, which causes blast disease of rice produced picolinic acid and piracularin as toxins. And the last one is biochemical alterations. It's been observed that infection of the host by the pathogens brings about biochemical change in the host which may prove toxic to the pathogenic microorganism and cause resistance to the pathogen. Production of certain new enzymes and other compounds are synthesized and accumulated in high concentration. This may also add to the resistance of the plant by being toxic to the pathogenic microorganism.
Okay, this is the end of our presentation. If you have any question, you can ask in comment section. So thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.